Synthetic biodegradable polyester polymers are an important class of polymer with a range of biomedical application, from drug delivery to advanced tissue engineering application, as well as cell therapy. Obvious examples of these polymers are polylactic glycolic acid, polylactic acid, and polycaprolactone. These polymers represent the industry gold standards owed to their tunable degradation and low cytotoxicity profile. However, the widespread translation into biomedical and clinical setting is hampered by the lack of cell adhesion moieties. There are a number of strategies that have been developed to introduce cell adhesion site into the structure of this polymer. These strategies include conjugation to a cell adhesion peptide, or physically blending with a secondary polymer, or in fact growing a second polymer from the structure of these polyesters. All of this to make them cell adhesive. All these strategies are complicated and challenging for direct translation into industrial and clinical setting. So we asked the question, how can we make these polymers cell adhesive without doing all this complex modification? To answer that question, we rendered the polyester polymer cell adhesive by changing one single atom. Here's how we did it. Here's the overview of the project to show how we made polyester polymer cell adhesive. We created two variants of the polymer. One has a new functional group called dimethylaminoethanol. The other one lacked the functional group. We created the two polymers using a ring opening polymerization. And then we have taken the polymer through a membrane emulsion process, essentially to create macroparticle. They have a utility as a cell culture delivery system or cell delivery platform. When you resuspend the two variants of the macroparticle at physiological condition, one will have a surface with slightly positive charge, the other one has more hydrophobic surface. And this is significant because when you mix the macroparticle with a clinically relevant cell type, for example adipose-derived stem cells, the cell can readily attach to the surface of the macroparticle, whereas in the non-modified variant of the polymer, you don't see any attachment of the cell to the surface of this macroparticle. And here's how we did the synthesis of these two polymers. We used a standard ring opening polymerization. We used two different initiators, one dimethylaminoethanol group, one propanol. We created the two different polymers, and then we created the macroparticle using a membrane emulsion technique, as I mentioned earlier. We then look at the morphology and size distribution. We wanted to understand if this modification of the polymer with the new functional group can introduce any change in the morphology or size distribution. We noticed that in either cases we are able to create near monodispersed macroparticle, and there was no significant difference in the size distribution, and the morphology of the macroparticle remained smooth and uniform. We then look at the cytotoxicity profile of these two macroparticles. We culture them with the adipose-derived stem cells, and we use a standard assay, LDH, lactate dehydrogenase assay, to assess the cytotoxicity profile. We noticed that in both cases, modified and unmodified, we maintain a low cytotoxicity profile, and there is no significant difference between the two groups. We also look at the qualitative assay using a live death staining to look at these to look at the cytotoxicity profile of these two polymers. Again, we confirm that there is no significant toxicity between the two groups. Importantly, we look at the cell adhesion at physiological condition. We created the two macroparticles, we resuspend them with adipose-derived stem cells at physiological conditions. We noticed that the cells can readily attach to the surface of this macroparticle. As you can see, they're making focal adhesion with the surface of the macroparticle, whereas the non-modified macroparticle cells will rather attach to the surface, to the solid substrate of the tissue culture plastic. And this clearly demonstrated that the modification of the macroparticle imparted the cell adhesion properties at physiological condition without further modification. We finally look at the degradation profile. We wanted to see if the introduction of the new functional group can introduce any change in the degradation profile. We noticed that there is a slightly faster degradation profile when we look at the new polymer. We followed this study for 21 days using the scanning electron microscope, as well as gel permission chromatography. Overall, we maintain the degradation profile of the polymer between modified and non-modified grade. So in summary, here we presented a new way to render polyester polymer cell adhesive at physiological condition without further modification, which can fit into the pipeline of a variety of clinical and industrial applications, ranging from drug delivery, cell therapy, cell culture to tissue engineering. We exemplify this by using the new polymer to develop a 2-in-1 microcarrier system for culture and delivery of clinically relevant stem cells. We've shown that this modified polyester maintains a low toxicity profile and desirable degradation profile whilst enabling cell adhesions. Finally, if you wish to access these polymers for industrial application, please get in touch directly with me or via UEA Business Office. Thank you.